Hello, Jonathan Wagner here from solidzymes.com, and today I'm going to show you how to do a Bradford assay to measure protein concentration. Usually I use the absorbance of a protein at 280 nanometers to measure concentration, but if you have a protein that's not purified, maybe it has DNA in it, or maybe it's a mixture of proteins, then the A280 approach doesn't work anymore. And today I have some lysate from E. coli. I need to measure the total protein concentration so that I can do my experiments consistently using the same amount of total proteins. And I have a Bradford assay kit with some convenient instructions. I'm just going to go through it with you really quick. So I'm going to use the platereader protocol. So I need just 10 microliters of a bovine serum albumin standard. I've labeled tubes here for each different BSA concentration. So I'm going to go ahead and put 30 microliters of buffer in all of the tubes that I'm going to dilute. 30 microliters of buffer plus 30 microliters of the 2x concentrated protein is going to make my standards. Now it's important that the buffer I'm using doesn't have any detergents in it, so there's no Triton X, no SDS, nothing like that, because those detergents will interact with the Bradford reagent and turn it blue, and we don't want that to happen. We only want it to turn blue when we add the protein. Okay, so this is the 2 mg per mil bovine serum albumin standard protein. Came with the kit. I'm going to put 30 microliters in this tube, which I've labeled 2 mg per mil. I'm going to get another 30 microliters and put it in the tube I labeled 1 mg per mil that already has 30 microliters of buffer. Pipette up and down to mix it, and then fully discharge the pipette. Pull up 30 microliters, put it in this tube, which is half a mg per mil, and so on down the line. And I'm also going to have a standard at the end that has no protein in it whatsoever. That'll be my blank. Okay, so my final standard is 0.125 mg per mil. I'm just going to put a little buffer in my 0 mg per mil standard. And now the buffer I'm using is the same buffer that my lysate is resuspended in, so it's similar to my protein. And now I just need to put 10 microliters from each of my standards into the plates, into the wells of my 96 well plate. Okay, so now I have 10 microliters of each standard in the plate. I'm going to put in 10 microliters of my E. coli lysate, and I'm afraid that this might be too concentrated. It might be even more than 2 mg per mil, so I'm going to go ahead and make some twofold dilutions of this lysate as well, and I'm going to measure 3 different concentrations to make sure that I get a sample of my unknown that's in the same range as my standard curve. Alright, so I have my 3 unknown samples and my standard curve. I'm going to go ahead and pour out my Bradford reagent into this trough so I can use the multi-channel pipette to start all my reactions at the same time. And once we start them, first I'm going to mix the samples for about 30 seconds, just holding the plate and tapping. And then I'm going to let the samples develop for about 10 minutes, make sure the color isn't changing anymore. And then we're going to read it in the plate reader. Now I do want to note that before you pour out your Bradford reagent, you need to mix the bottle, and that's very important. Not everything in here is completely soluble, so if you just take the material off the top, and everybody in the lab keeps taking the material off the top, eventually you get to the bottom and your results are not going to be the same from the old bottle with a little bit at the bottom. And as soon as you get a new bottle of Bradford reagent, everybody's going to freak out because your numbers will be different. So just make sure that everybody in the lab is shaking the bottle before they pour out their reagent. Alright, so I'm going to add 200 microliters of Bradford dye to my 10 microliter samples. Mix it for 30 seconds. Just by tapping on the side of the plate. And I don't know if you can see that, but the dye is starting to turn blue. The Kumasi in the Bradford reagent interacts with the proteins and it shifts from a reddish brown color to a blue color. So now we're just going to read the absorbance of all these wells at 595 nanometers. And then we're going to plate it, we're going to graph the results in Excel, and I'll show you the results after this. Alright, so I've imported the data from the plate reader into Microsoft Excel, and I'm going to show you how I made a standard curve and calculated the concentrations of my unknowns. So the raw absorbance data is here for my BSA standards. The first thing I did was subtract the sample with no BSA from all the others to get an adjusted absorbance over here. And then I made a graph using the known concentrations and those absorbances. You can see the graph is not linear, it's curved. So we're going to have to do a little bit of relatively complicated math to calculate the concentrations of our known samples. So in order to do this, you just click on the data point, you say Format Data Series, you say Add Trend Line. Choose a trend line that fits the data pretty well. In this case, I'm going to choose a polynomial, and I'm going to say Display Equation on the chart. Alright, let's make that bigger. Alright, so here's the equation, and there's two ways to calculate the concentration of your unknowns. What you can do is put the coefficients from the formula in 3 different cells, like I've done here, and then you can write the formula for y here in another cell. You can see it's equal to coefficient 1 times this cell squared plus coefficient b times this cell plus coefficient c. And then what I can do is I can start putting in values for x, and it will calculate the absorbance that I would have got from that. So let's say my protein was 1 mg per mil, then it predicts I would have an absorbance of 0.39 based on this standard curve. And I could just keep on guessing until I get the absorbance that matches my unknown, and then this would be my answer right here. Or I can let Excel do the guessing for me. It's much faster. So I can use goal seek. So I've copied the same section down here, and this time I'm going to let goal seek guess my X values. So to do that, click on the cell with your formula, go to Data, What If Analysis, Goal Seek, and we're going to set that cell equal to one of the values for the absorbance of our unknown. So let's try 0.407. That was the first one. Okay, and we're going to get that by changing this cell over here for X. Okay, so start guessing. It guesses that a value of 1.02 is going to give me that absorbance. So 1.02 mg per mil. And then we're going to repeat that for the other two unknown values that I have here. So let me just pull over my spreadsheet where I've already done this. You can see here are the two other values that I got for X. And if I multiply by the dilution factor that I used when I was preparing these samples, which is 1, 2, and 4, these are what it predicts the concentrations of my total proteins are. And you can see that the first one doesn't quite match the other two. It's about 30% less. If you look over here on the graph, you can see that for an absorbance of 0.4, that's up here, it could be here, or since the line starts to go back down, it could be over here above 2 mg per mil. So the line is not doing a great job of predicting values out here above 1. So I'm going to just focus on the diluted samples down here where there are more data points. And you can see both of those match pretty well, 1.3 mg per mil. So I'm going to take that as my total protein concentration, and I'm going to use it in future experiments. I really hope that you enjoyed this video, and if you're interested in what we do here at SolidZymes, come visit us at solidzymes.com. We offer enzyme activity assays and enzyme production services. We'd love to have you visit. Thanks for watching, and I'll see you next time.